So before we jump in, we had a presentation from Fazlin and Najafi on Friday that we share on YouTube. I will be curious to hear what people opinions are on it, because I think it's the results from this study is putting in questions the amount of temporal predictions that are present in visual cortex. So I think it's relevant to some of these experiments that we're doing. So you can find the video on the YouTube channel. It's the meeting that is listed here. And if you want to discuss it or provide your opinion, just click on this link here. It's going to create forum to discuss the presentation. And then I will be really curious to hear opinion on it, because I think it has a number of complementary experimental design that I think are important. Especially, I'll be curious to hear Jeff's opinion and Sarah's opinion on this presentation. Okay. Let's get started. So good morning, good afternoon, everyone. So as usual, this meeting is going to be recorded. So if anything occur during the meeting that you would prefer us to edit, please let us know at the end, and then we can edit it before posting it on YouTube. Okay. So the meeting agenda is on as usual on this page. So what we are planning to talk about is first share an update on data collection for the SLAP2 platform. Then Jessie is going to share some of the analysis that she did on SLAP2 data, and then Lucas also was scheduled to present his update. Anything else we should discuss today that is important? Okay, so we've been working very hard with Andrew, Made, and Casper on collecting data for SLAP2. And so our current plan for the next 6 months is highlighted in this Google Sheet that I'll share in a meeting thread. And so what you're looking at here, I don't know if I can get into can you see this sheet well? Or?
Yes.
Okay. So what we did in the last few weeks, you can see on the schedule, is we finished what we call SLAP version 2. So this is the iterated design that we mature over the summer based on the first pilot we did before in the spring. And so we added 4 experiments, so four neurons to this data set. So we're probably close to 10 neurons at this point. And what we're going to do is package this data set as efficiently as we can and share it with all of you. We think that should be sufficient to finalize any analysis that you have started on this data set. If more is needed, we can certainly consider it. Then for the next iterations of SLAP2, what we are planning to do is to go through the same experimental design that we developed for the mesoscope and the NeuroPixel dataset. So what I'm going to share here is a slide that highlight this goal. So can you see this full screen slide? So you might recollect that we are currently planning to have 2 cohorts of 2 photon data and 2 cohorts of NeuroPixel data, and then the intent is to have a sensory motor coupling at the start and on one end, and on the other hand, a sequential coupling. So this will be more similar to work done in Kelly's lab, and this will be more similar work done with Andrew Bastos and Alex Meyer as a training. And then, yes.
Sorry to step in, but I think we're seeing the wrong screen. Oh. We just see the click to exit full screen.
Okay, so maybe
let's
stick to this view. Probably should. Okay, so you see the 2-photon cohort here as this. So one is the, as I said, sensory motor coupling, and the other is sequential context. And then you switch from different context later on. And the NeuroPixel cohort is mirror. Now, SLAP2 has some bandwidth that Casper kindly gave us to run. And so what we thought will be really impactful is to do a sensory motor cohort on the SLAP2 platform. And so that means exactly the same experiments that are done on the 2-photon and NeuroPixel. So this way you can compare data set acquired across the brain with NeuroPixels, with dance recording, the mesoscope, with calcium on the SLAP2 platform, where we are looking at a single cell. So that's the intent. And to maximize the impact, what we are proposing to do is do this designs with 5 mice with glutamate imaging. So this way we just focus on measuring the input to one cells during all of those sessions. So that means imaging the same cells across 4 sessions on SLAP2. So in total, this will be 5 times 4. So 20 sessions worth of data. And then that will be the first 5 mice. So it will be collect. And so in this sheet, you can see the plan, because we have to fit this session into the schedule over the year. And so we take into account Christmas and all of those breaks that occur here. And so that means we probably would have done these recordings in early January if everything goes well. And then the intent is to switch midway to do voltage imaging across the dendrites. So this way, because it's currently challenging to do both at the same time. And so we would record the voltage with a recent voltage sensor across the dendrites in this paradigm. And so that means similarly, the schedule will drag, because it takes time to train all the mice and do all the process until April. Are there any questions? Yes.
Yeah, just to clarify, when you say voltage imaging, you're not meaning regular 2-photon. This is still like SLAP2 that you're looking at dendrites or one cell.
This is, and Casper and Andrew can weigh in. This is using a recent sensor that currently we can't reveal the name, but this will be voltage imaging across the dendrites of the cell. So
specifically, it'll be voltage imaging in 2 planes in any given session. We can discuss this if it's really useful to do it in 3 places. There's a very different strategy we could use, but yeah, 2 planes.
But 1 neuron at a time.
That's right.
1 neuron at a time. Yeah.
And the same neurons across all 4 sessions.
And can you tell the type of neuron? Can you do optotagging?
So this will be a pyramidal cell.
This is it. I mean, it's imaging. So anywhere that you could optotag, you could just look at the cell. Like we can target, but we're imaging layer 2, 3, CAMK2 positive. We can choose broad tufted or slender tufted, or or where the cell body is. But and maybe we should do that, but pyramidal cells.
Karim?
Just to clarify, we cannot do this in the same animal, right? So it's either voltage or glutamate.
That's the current limitations of the platform, and I think everybody wants to do everything at the same time. But I feel like with the current set of technologies, this sort of will give you very, very good data on both modalities.
Yeah, I'm not questioning that. It's just the ability to analyze the actual input as processed by different parts of the dendrites.
That's right. But that will be it's a limitation of that study. But I think the intent here you see is multimodal recordings across like NeuroPixels, mesoscope. So you're going to have a lot of data on the same experiment. Okay. But yeah, Lucas.
Yeah, just related question. It seems like we're kind of skipping calcium as a potential third sensor. Is there a reason for that?
Well, we can discuss that. Calcium is going to be recorded in the somas with the mesoscope. So for the mesoscope is going to be pan-neuronal. So we're going to record both excitatory and inhibitory neurons in V1 and LM. So you'll have a proxy there. It's true that you're not going to have it's currently challenging to do all of these at once. But calcium across the dendrite, is that what you meant?
That's what I meant, yeah.
Yeah. The plan could change on the way if something really amazing come up. I'm sure Casper has some thought there. In my view, this is like a trade-off. Like strategically, what is the most important? And I feel like there are a lot of predictive processing theories that have advanced the role of voltage in those dendrites. And so I feel like that will give something that actually we don't have today.
Yeah, that makes sense.
I just want to say that, I mean, we recognize that at least glutamate and calcium for input output is very, very valuable. And I think we will try it for these experiments. But we had trouble with it in the past. A recent batch of animals has had really good calcium imaging during glutamate imaging. Interestingly, we don't know exactly what the difference between those is. We're still learning. We might be able to get calcium. We just don't want to promise it.
Gotcha. Yeah, thanks.
Are there more questions on this plan? I want to highlight two people here in addition to Casper is Andrew Shelton and Made, who are you see the name in the schedule. They are committed, and I'm really thankful for all the work they're planning to do. It's a lot of sessions. So you're talking about 40 sessions distributed over the years, and they're doing this in addition to other projects that they have at the Institute. So thank you. Okay. Any other questions? I think we can move on to the analysis unless there are more comments.
A quick one.
Yep.
So we're talking about which data will be available and so on. We were just wondering, a lot of the August sessions on this one subject. So the 25th of August, that session worked well, but the other data are not uploaded yet. And I just wanted to ask if it's just the packaging or if something in the experiments went wrong so that they do not make sense to be analyzed, or just wanted to ask what the status is there.
It's exactly kind of feedback we're looking for. If there's an issue, we should fix it right away. So maybe that's in the next section of the discussion.
Yeah, I can write it down and we can follow up there. Yeah.
Okay. Yuji, are you here?
Yeah, I'm here. Hi.
Hello. Why don't you present yourself and then and share your screen?
Okay. Yeah. Hi. So sorry. Give me seconds.
First, thank you for presenting. I think it's very late in China right now, right?
Yeah, but it's fine. Yes.
Okay.
Currently stay alone, so it's fine. So
thank you for accommodating.
It's my pleasure. Thank you. So can you guys see my slides? No.
Yes, thank you. You might want to go full screen.
Yeah, give me. So is this? Yeah. So can yeah. So yeah. So this is a very preliminary kind of summary of my current analysis and finding. And so why we choose PCA is I think the SLAP2 data are high dimensional, and I think PCA allows us to simplify these complex and high dimensional signals into a few orthogonal components. that capture most of the variance so that we can identify the dominant population level patterns. And the second reason is PCA provides a way to separate different types of variance, allowing us to understand the meaning of each component, such as whether it corresponds to global activity or condition-specific patterns or other task-related information. And currently, our main finding is PCA5 may have captured a flip or reversal in the activity patterns from control 1 to outbook and control 2. And this flip could be, might be related to adaptation, but I still need more evidence from other studies and more analysis to be sure. So, and some components capture other meaningful aspects of the dynamics. And well, I'll start by sharing the overall approach, how the analysis was done, and the results we found, and some possible interruptions. Well, please feel free to interrupt me at any point if something seems unclear or if I've misunderstood something. I'd really appreciate for your guidance and any suggestions. Well, overall, there are 4 main parts or steps. Specifically, we flattened the ROI plus time matrix into a one-dimensional feature vector and then run PCA on this vector. And this decomposition gave us 3 closely related quantities, components, the loadings, and PC scores. The components say pieces in the lettering showing are the new axes that capture dominant patterns in the data, and the loadings which tell us the spatial temporal structure of each component in terms of the ROI and time, and PC scores. And I think basically showing how much each child expresses that component. So our first check is like, are these pieces explaining real shared variance, or it's just fluctuations? So after obtaining the component, we calculated the explained variance, and we compared the we compared them of the real data with shuffled non-distribution to test whether PCA really captures shared variety patterns across ROIs and time. And rather than the independent fluctuations. And for current results, we focused on the first 5 components. The green dashed line here in the plots show the 90% quantile, and the red line represents the 100 value. Well, like these 5 components were from our initially tried with only the trials from the Audible blog. At that time, when we used the 99% threshold of the non-distribution, we found that there were more than 100 significant pieces above the threshold. So we decided to concentrate on just the first 5 components for further analysis. But according to our latest update, we include the trials from both the 2 control blocks together with the Audible blog. And there are around 10 components selected. But currently, we haven't systematically looked into the rest of the components yet. So the results today are focused on the first 5 components. So going back to the first 5 components, and we also plotted to say how much of the variance each accounts for across both 2 plans. Each of the first 5 PCA shows a higher explained variance than the shuffled data, indicating sorry, excuse me?
I can hear you, Jesse. It sounds like someone just leaked in. I don't know. Can you hear me?
Yeah, I can hear you. Can you hear me now?
Yes.
Okay.
It was very clear to me. Your talk was very clear to me, Jesse. I feel like you can continue.
Yeah, thank you. So we also visualized the cumulative explained variance of these 5 components for both real and shuffled data into plans. And here the yellow line indicates the shuffled data and the blue line represents the real data. I think this component, I think these figures together confirm the top 5 components already capture the dominant structure variance present in the population activity. So next, to understand what these components actually represent in the data, we looked at their loadings. As we mentioned at the very beginning, we flattened the time plus ROI matrix into a one-dimensional feature vectors to run PCA, which give us two important outputs in addition to the components. Well, we found that for the loading parts, we found that all components are stimulus locked. And temporarily, PC1 shows somewhat global patterns, and I think many ROIs share the same loading sign within a specific pre-stimulus window. And I think I think this suggests that PC1 captures a dominant shared variance structure, like a global co-fluctuation mode expressed before stimulus on site, and PC2, in contrast, has broadly negative loadings across many ROIs, almost a mirror image of PC1. And PC3 shows temporally specific late loadings with stronger value emerging later in the post-stimulus period. And PC5 shows a transition from early positive to negative, then rebounds to positive around 0.2 seconds after stimulus. And specifically, different ROIs show component-specific weights. Like, for example, the ROI 27 is carries stable positive weights across these 5 components, and while ROI like around 80 shows opposite signs, specifically with positive in PC3 and negative in the PC2. So I think this results means PCA extracted task-aligned structure rather than ongoing stimulus on related fluctuations. So then we focused on the selected pieces scores and calculated their the correlation the R values of these PC scores with child index, and I think these scores tell us how much each component is expressed in given trial, and by plotting them across the session, we can see whether these patterns shift over the sessions. And here's the results for DMD1, and we tested whether any components show systematic change across trials. The positive R value here indicates that the pattern represented by the component gradually strengthens over the session. And in contrast, the negative R value means that when larger absolute values are, the component's pattern straightens in the opposite direction. And in practice, we think PC3 and PC5 showed the strongest positive and negative trends, respectively, with PC3 at 0.19 and PC5 at negative 0.44. And I think PC3 shows a slightly positive calculation value, might suggest a weak upwards tendency over time, and PC5 scores gradually move to the negative direction across trials. And however, PC1 and PC2 remains relatively stable trends across the trials. And then we followed the analysis framework of Lucas, which specifically investigates how adaptation depends on the on stimulus orientation. With question which stimulus orientations are mainly driving the activities or patterns. Specifically, we divided the entire session into multiple sub-blocks in which control blocks were subdivided into 16 and the audible blocks into 4. And we then compared the PC scores across sub-blocks using the sub-block as minimum unit. And in these plots, the red bars correspond to the red bars is the audible block and the gray bars are the control block. The findings, today's findings, I'll focus on this three as I'm still didn't understand the other components clearly. So for example, the PC1, I think the PC1 captured a dominant orientation tuning pattern, and it shows similar response to 0 and 45 degree, but showed a clearly distinct response to 90 degree. And the PC3 likely reflects a collective response pattern of ROIs that are particularly sensitive to the 45 stimulus. And we can see that in the audible condition, their response are stronger than in the controls. And finally, PC5 seems to capture a reconfiguration of the population response at 0 orientation from control 1 to audible and the control 2. I think this.
Jesse, I have a question.
Sorry.
Yes. Well, the value of this analysis is to extract meaningful variance together and see when this component is very much engaged in across the spines or the synapses. And have you looked at separating the audible trials from the non-audible trials? The standard trials, because I think these are still within blocks, right?
Yeah.
Because I wonder if seeing whether to actually test this idea that the PCA is extracting specific mode of activity related to audible presentations. I feel that might be helpful. Because here it's all blocked together. Is that right?
Yeah. I'm trying to do that part, but I didn't finish.
Okay.
Yeah, I didn't finish it as recently I'm trying to understand like what this part of results tell me.
Okay, can you explain it again? Yeah.
It looks like the main effect that's captured is the orientation tuning, right? At least if you look at component 1 versus 2, maybe they seem to differ mainly in terms of what orientation you show. So maybe that's the most strong, that's where most variance lies, which ROIs you engage for which orientation or so. But maybe I also understood this wrongly.
I do have a comment as well. I think this analysis is somewhat similar to a analysis I did a little while ago, making use of PCA and looking at orientation tunings as well. So I can include a link to that in the chat, and perhaps at another meeting in the future I can see how my analysis relates to yours, because I think there's a lot of overlap.
Yeah.
I have a quick question. And my internet is bad. I'm sorry if I interrupt everything. Can you hear me?
Yes.
Yeah.
So because I might have missed it, did you look at what the components look like? Did you plot them, especially for the DMD1 and DMD2, whether they are the same? If you say principal component 1 for one plane or the other plane, and you look at the component, you look at what it looks like. Did you look at that?
Yeah, but I think they're different.
You had them in the previous slides, right? Can you show that to Alex?
Yeah, like
no. Yes, here.
yeah.
I mean, you need to zoom in, maybe.
Oh yeah. Give me a second. So is that this part or?
I think the left figure with the image. I think this is what because you're showing the loadings across space, across ROIs, and across time in the trials. This is this left part, right?
Can you see that clearly?
Is it the right hand side, the plots with the red and the is it the time series or is it the 2D plots?
The 2D plots. As I understand it, time here is aligned to the 0 is the presentation of the gradings, the drifting gradings, and the y-axis is all the ROIs. And so a stronger pixel means the PC's weight is strong on that particular time and that particular ROI. Is that correct,
Oh, you
Jesse?
did the PCA. So the PCA was done for the entire data. You didn't do the PCA per plane. Is that correct?
The first column is the plane 1, like DMD1, and the second column is the DMD2, the second plane.
But the PCA, did you use all the data or did you have different PCAs for different data?
I used it. Makes sense too. I applied the PCA to different data, like the plan 1 and plan 2 separately.
Right, and so then my question is the result very similar or is it very different? If you can plot the first component and second component, you can look at them in a plot, right?
Yeah, but I think they're different.
Well, I mean, for instance, if you look at PC3 here on the third row, the patterns that is extracted across the ROI is actually similar. You see, like these PCs are typically flat until the drifting gradings occur, and then they go up and they stay high for the rest of the presentation. And this is true both for DMD1 and DMD2. In this particular, you see what I mean? The third row. But now, if you go to the fourth row, that's not true. That somehow the statistics of the data in you have a burst of activity that is captured by PCA, and on the other end, it's negative. It's going down. And that could be due to how PCA work, right? It has a sign uncertainty whenever it recomposes, because you can assign a weight to the components or to the trace. So you have to be careful with signs here.
But the
Oh.
components are different for the
Sorry,
different.
I'm in a meeting.
The
currently,
components are different
but
for the different
next time.
planes, right?
I'll be
You have
back at
different- you have a-
4 or 3.
You have a different component for one plane and a different.
Right, Jesse?
Sorry, I didn't hear. It's freeze here.
So you have two PCA spaces in a way. You have one PCA space for the first plane and another PCA space for the second plane. Is that correct?
Yes.
I think Alex is suggesting that we do an analysis where both planes are concatenated so that you could compare the components across both planes.
Sorry. Thank you. Yeah, I was considering this, but the point is like as there are the number of ROIs are different from these two plans, and so currently for this first step, I use two PCA on different plans to say sorry.
Yeah, it's a good point, but then we cannot compare the planes because they're in different PCA spaces, right?
Yeah, I'll do that next step.
And it may also be informative to plot what the components look like. If you plot the first principal component or second principal component, we could learn maybe what they look like. For example, if they just go up and down for one of the blocks.
Yes, yes. Yeah, I will include that part in the following analysis. Thank you.
Okay, Jesse, do you want to return to your presentation? I don't know, you were on a later slide.
Yeah, it's so yeah, so let's go to like the yeah, so currently I think this this is the only results that I find from current analysis when I combine different parts together. It looks like PCA5 encodes, but this is only for DMD1. As like especially for DMD2, the PC, especially for PCA5, it's act different in DMD1 and DMD2. So like PCA5, I think it encodes processing sequence like early response, mid phase inversion, and late update and stringent, and this pattern shift emerges. This pattern changes after the audible emerge, and stringent across the trials, and the sub-block plotting confirms these results. But like we can see that it's clearly that like some ROIs are strongest than the other, and at some specific time points. So whether this kind of pattern was driven by specific ROIs, like ROI 27 around 200 milliseconds, is a good question. And so this is my current work in progress. The first one is literature review to make sure that or what kind of information PCA5 represent, and like and so on. So yeah, this is
very good. Sarah made a point in the chat about one of your next step, which I think mirror your thinking. So I think you said it will be. She said it will be interesting to see which ROI contribute to PC5, and so I think I agree with her that plotting the weight across space in the way that you visualize the dendrites. And if you could also do that across the two DMDs, you will see how the extinguishing PCA from the first block to the next block is distributed.
Mm-hmm. Yeah.
That makes sense to me. Any other? Thank you, Jesse. Any other questions? And well, you have already had some really good feedback to work with.
Yeah, thank you.
Lucas, do you have things you want to share?
Yeah, let me share my screen. Okay, so let me know if this works.
Great.
Yes, perfect.
Yes.
Okay, yeah, I'm very happy to give a brief update on the on our results on contextual modulation of synaptic responses. We had a lot of fun analyzing the new data, mainly because we have Levke, who joined our efforts here, who's an intern in our group, and did the analysis, and will also represent these results in the second part. The data showed very strong effects, so really looking forward to hear your thoughts on that. Before we go into the new data, I would like to just give you a brief heads up of what we've been looking at previously and what's kind of the type of analysis is that we're interested in. And we're interested in contextual modulation during the oddballs. And this is a figure from John Ham's lab, where the idea is in this oddball paradigm that we compare responses to the same stimulus, but under different predictive contexts, or different temporal contexts. And so the idea is that one shows an oddball where you have the deviant response shown on and on, over and over again, and then you either look at the same stimulus as part of this redundant sequence, or as part of a deviant oddball, or in a control condition where it occurs randomly. So we have three different conditions, and the question is, how does the different contextual context modulate these responses? And here we will also later on look at these aggregated data. So what's shown here is this is somatic ROIs, so soma imaging, and here the average response of these somas is shown for the control. And then this is a very classical finding that you find the redundant condition to be suppressed relatively in response, and the deviant response to be enhanced. So the question is, do we see the same thing for the synaptic ROIs that you guys provide in the SLAP2 data? So previously we were analyzing this stimulus design. I think Jesse also showed data from these previous experiments where we had one control block where these orientations would occur at random. We had the second block, which is the oddball block, where you see the 0 degrees, the standard, which is then occasionally violated through either 45-degree or 90-degree. And then there's the third control block that's essentially the same as the first, but it comes after the oddball. So we use that to essentially repeat the analysis that John did in his previous paper by taking all ROIs that we found to be responsive for a given orientation. So here it's 0 degree, that's the standard, or 45 and 90, so the two deviant stimuli. And you can see in gray the average response in gray to the control condition, and then in blue the redundant condition. And you can see there's a bit of a suppression here, whereas kind of a deviant enhancement, we only saw indications for that in the 90 degree orientation, potentially because 45 degree is closer to 0 degree, which has been shown repeatedly during that oddball. So this was one type of analysis that we did, but then we also looked at the course of responses over time. And here we applied an additional trick, so to speak, where we looked at responses that were only responsive during control 1, or required to be responsive during control 1, whereas here we selected them if they would be responsive during any condition. And what we picked up with this selection was synapses or ROIs that were strongly responding in the beginning. But we figured out that there's a bit of a transient. So because was no habituation in that experiment, these synapses would decrease their responses over time, then have a low activity level. And then we also observed a bit of a recovery from potentially synaptic adaptation, which was similarly observed for the 45 degree, but not for the 90 degree. And then we also selected a different set of ROIs that would be responsive during the second control. And these had a very different behavior, because initially they were hardly responsive. But then after the oddball, they would become active, which is very interesting, indicating that there might be some form of plasticity or so. Maybe those are silent synapses that then become active. Just some speculation here, but to take home, by selecting these different sets of units responsive either during first control or second control, we saw very different behavior. And this is just an example of an ROI that only appears to be responsive in the second control. So in the lower row, you see the response of the ROI in the light gray for the first control, and then during second control gets this very interesting response profile with an kind of early and late response, or an off response potentially in the 0 degree or 45 degree, and then kind of an early response in only for 90 degree. I think this is just an interesting example to see there are definitely changes in responsiveness. So in addition to contextual modulation, it's something that one can pick up with these kind of analysis. So what we concluded from those initial experiments was that we see that for ROIs that are initially responsive for oddball stimuli, that there's a depression and potentially synaptic depletion for the standard stimulus, which might sweep over to the 45 degree deviant. By analyzing the kind of transient things, we initially thought there was also long-term depression, but it seemed to be more of this transient effect. And then for ROIs that become responsive during the experiment, so after the oddball, we saw a mild enhancement for deviant context, and there seemed to be long-term potentiation for the standard stimulus. And yeah, just based on recent work of us, we kind of saw analogies to synaptic depletion, where it seemed that the first control was higher because of the initial exposure, so there was no habituation. Synapses appeared to be more responsive in the beginning of the experiment, and then in the second control block, low responses could be caused by a slow release from synaptic depletion. So moving forward, the new stimulus design new stimulus paradigm was designed to mitigate this. So what was added here was kind of an extra buffer to account for these, let's say, transient effects. Actually, I think in the orientation 1 block, it's even more. It's double the size to account for any habituation effects. So extending these helps us to, look at when do these responses become non-transient and factor out these effects. And then maybe even more importantly, this stimulus design contains a second oddball condition where we flip the standard and the deviant orientation. And this allows us in this context to study the same stimulus under 3 predictive contexts. So under the random control, redundant and deviant for the 0 and 90 degrees. And the 45 degree we can observe under different predictive contexts. Yeah, so with this, I would hand over to Levke. Maybe you can briefly present yourself, and then, yeah, we talk about the new results.
Yes, hi, can you hear and see me?
Very clear.
Perfect.
Nice to meet you.
Okay. Yeah, nice to meet you guys too. Thank you for letting me join. Really exciting data and exciting community. I'm hearing myself double somewhere.
Maybe that's me, probably. Sorry. Have to find the right window to because I'm sharing. It's a bit tricky to unmute.
Okay. I think it's fine. Okay. Perfect. So what we first, my camera seems to not want to stay on for some reason. You've seen me. I'll just continue talking. So what we're looking at here first is the aggregate plots during like the conditions that Lucas just described. So we can see the 0 degree in the first plane, which is the DMD1. Sorry, which is actually superficial in this experiment, so it got switched around. So the 0 degree stimulus, which is in the control blocks. So we have now 3 control blocks. Just one of the stimuli presented with the with the regular frequency, and then in the first oddball block, the 0 degree stimulus is redundant, so presented over and over again. And in the second oddball block, the same 0 degree stimulus is the deviant stimulus, and you can see that we really nicely see that in the redundant setting, the activity is lower than the in all of the control blocks, and in the deviant setting, so where that is deviant, it is actually higher. And you can see that consistently also in the other plane, and also with the 90 degree stimulus, which is basically the same setup. And then you can also see that in the 45 degree stimulus for where it is deviant during both of the oddball paradigms, you can also see heightened activity for the deviant stimuli. So kind of what we would expect for heightened activity according to or the representation of an unexpected stimulus. And so what we now wanted to do was look at the same timing evolution that Lucas looked at with the earlier data. And so this first is also an aggregate plot for all of the ROIs that were responsive during any of the control conditions. And what you can really nicely see here as well is the kind of depression of activity that is going on when the redundant stimulus is presented, and then the heightened activity in the when the deviant stimulus is presented. And maybe what you can kind of like infer. So we will continue to do, this is all very preliminary data, but so we will continue to do statistical testing. But what you can maybe see is that the response to the 45-degree deviant stimulus, the second time it is presented as a deviant stimulus, might actually be a little higher. But what you can't see is the things we observed in the past experiment of like the kind of transient effects that we saw earlier. So this seems to the activity of the RIs during the control or tuning blocks seem to be quite consistent over the 3 blocks and over time within these blocks. And when we now look at these temporal evolution plots, kind of step by step and differentiating which are synapses we look at. And we first look at all of the synapses that are responsive during the tuning 1 block, so only during the control 1 block. You can also kind of see fairly stable like responses during the control sessions. Maybe in the 45-degree stimulus, you can see some sort of depletion going on, like overall activity of these RIs that are responsive for the 45-degree stimulus is kind of lowering throughout the whole process. And we can compare this to looking at all of the units responsive during tuning 2. Where you can also kind of see this kind of heightened activity during the second presentation of the 45-degree stimulus. And when you look at the 90-degree stimulus, especially during the tuning 3 specific RIs now, you can kind of maybe see some sort of depression of activity in the third or in the last control block. So when you look at the 90-degree plots, the ones on the very right, you can kind of see that the activity goes down in the third control block over time. And this is also visible, Lucas, if you go to the next slide. And this does seem to be kind of specific for the RIs that are responsive for the 90-degree stimulus, but that is something that might reflect this like, kind of lesser activity during the last control block. You can also see in the aggregate plots. So all of this is just like the first analysis we did with one of the samples available, and we're looking forward to include more data as it comes in. Lucas, do you want to do that? Should I continue?
Yeah, maybe it's easier because I'm on the one pressing the
yes
to take over the summary. As Luke was saying, we just have this one experiment. We couldn't. It seemed that the NW-B data for the other recordings from the same subject were not there yet. Levko also looked at the recordings from July, both of a parameter cell and a
VIP neuron.
A VIP, but some of the responses were not strong enough to the responses to be detectable to the different stimuli. So it seemed that we couldn't use these data. So I'm curious to hear what others find there, but somehow we didn't manage to pull out.
So
something.
Are you saying in the VIP cells there are weak visual responses? Is that what you said?
Right. It looks
Okay.
that we have problems detecting any difference from the baseline.
I see. It might be worth that we plot the receptive field using the. Yes, at the end of the session, we have receptive field mapping, and so it's possible that somehow we were not rightly aligned.
Yeah, so the data just seemed to be really kind of noisy overall. So there was no real, like no selective ROIs responsive for any of the stimuli. So it was just very general. Yeah.
Okay.
Yeah,
I mean,
that's a
it's
good
likely that
suggestion.
VIP cells have a different response profile too, right? So yeah.
Right.
And
Yeah,
did
but
you
for the
encounter other issues with the NWB files beyond responsiveness?
No, the loading was fine. So we could load and open all of them, and they looked okay. So at least the ones from July and the one from August. So this is the 25th of August experiment, and the others are not online yet.
This analysis that you presented is on one neuron?
Yeah, it's just one.
Okay.
The 25th of August. Yeah.
Okay. Because at this point we have, I think, close to 10 recordings. So and there are 4 more that we need to upload that were recorded in the last 3 weeks with Andrew.
Right.
So that should allow you to do some more statistics.
Yes.
That's great. Yeah. And we were kind of encouraged. This recording looked really great in terms of signal to noise ratio and how different these responses were. So I think this is an encouraging example, and then we see how it goes with. I guess there's also high variability between cells to be expected, but yeah, this one was really nice. I mean, we really saw the suppression when redundant, enhancement when deviant. And also, I mean, although we have to do the statistics, the predictive context seems to matter for 45-degree stimulus, very preliminary looking, like first glance. Interestingly, here we didn't see the clear transient effects visible in this experiment. So if we find the same thing in the other recordings, then it might we might be able to save recording time and doing the shorter controls again. There are some indications of plasticity, so some RIs might showed some depression or potentiation after the ball blocks, but we couldn't see this very clear difference by selecting units either responsive for first or responsive during first control or second control. So we think this might be very ROI specific. So next steps would then be to really analyze individual ROIs as well as their spatial relation. So can we, when plotting them in the imaging planes, do we see any clustering or so of units that show particularly strong deviance responses, or show changes in their responses to different orientations? And here I think it would make sense to team up with other groups here, for example, Sarah or Nicholas' analysis that were more refined in, would resolve the responses in the entire orientation space, and see how these change depending on upper response and how this relates to what we see in terms of contextual modulation. I think that would be very cool. So happy to hear what the others are doing, and maybe we could integrate this. Yeah.
Thank you so much. This was a great first pass at the new data.
Thanks. And thanks to Liv.
It's fantastic that you see a similar effect in the first session. So that means potentially we can leverage both types of session in the analysis. So we are 10:05. Would you be okay with sharing your presentations on the link on the forum?
So what
it's already there.
It's already there? Okay, fantastic.
So we opened the discussion for this meeting and put the presentation there. I don't know, is that what you were thinking of, or?
Yeah, in the forum. So if you go on the meeting day, there's a discussion, and then if you click on the link at the bottom, you just create a thread. If you already have it, that's fantastic. And maybe we can continue the discussion in writing form, because I think some of the key people had to go. and then we'll see what we do next week based on this discussion.
Perfect. Yeah.
But I think it's 10:06. I think we'll need to break. Thank you so much. Thank you so much for doing all of this analysis and sharing it openly. Thanks, everyone.
Thank you. Bye.
Thank you. Bye. Bye.